Hello students, welcome to the EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about the types of lipids from the paper Lipid Metabolism. The objectives of this paper includes how many types of lipids are present, what are their complexities, how it is significant, and how they act in a system. According to Blohr, who is a German biochemist, lipids can be best understood under three categories, and these categories are simple lipids, complex lipids, and the derived lipids. The simple lipids are those lipids which is a saponifiable lipid. It is also known as storage lipids, which two types of component, that is the alcohols and the esters of fatty acids. Another category in simple lipids are fats, which are glycerol with esters of fatty acids, and sometimes they are solid at room temperature due to saturation of carbon atoms. Again, oils are also a type of simple lipids which is fat in a liquid state at room temperature, and this is due to the unsaturation of carbon atoms. Another category of simple lipids include waxes, which are high molecular weight monohydric alcohols with esters of fatty acids, and these waxes include the true waxes, the esters of cholesterol, and the esters of vitamin A and vitamin D. So the types of lipid include the simple lipids, the complex lipid, and the derived lipids. This is what we have already discussed in the classification based on hydrolytic phenomena. Simple lipids are the lipids which contains glycerol as well as fatty acids. The examples of simple lipids include fats, which contain the saturated fatty acid, the oils, which contains the unsaturated fatty acids, as well as waxes. So the subtype of the simple lipids includes fats, oils, and waxes. Another category or another type of lipid is the complex lipid, wherein the lipids either contains the phosphorus group or it contains the glycolipids or the saccharides, or it contains the proteins, and henceforth the subtype of complex lipids is referred to as phospholipids, glycolipids, and proteolipids. The third type of the lipids is the derived lipids, which is either hydrocarbons, steroids, or vitamins. The complex lipid, which is also known as conjugated or compound lipids, are the fatty acid esters with groups in addition to the fatty acid, an alcohol moiety is present. Being extensively distributed in animals, bacteria, and plants, these complex lipids are the major component of the cell membrane and also found in circulating fluids. Complex lipids can be further subdivided into three main subtypes. The first is the phospholipids. The phospholipids are the lipids which are with a phosphate moiety. A glycerol moiety is also present, and an amino or a fatty alcohol with or without one or two fatty chain. Sometimes one inositol group, two phosphate group, or four fatty chains are present. The phospholipids frequently have bases with nitrogen and other substituents. Hensing in 1719 first reported the occurrence of phosphorus in brain tissue, followed by its extraction with ethanol, which was discovered by Vauquelin in 1811. Later, similar substances were isolated from brain in alcohol and which were further christened as acide cerebrique, cerebrotum materie blanche, or oleophosphoric acid. Goblet in 1850 isolated lecithin, which is also synonymously known as phosphatidylcholine, a phosphorus containing lipid from yolk of egg and brain, and designate it as lecithin. In Greek, the lecitho means the egg yolk. He also revealed that glycerophosphoric acid may possibly be brewed from lecithin. In 1868, parallel to the work of Goblet, Strecker demonstrated the presence of choline in bile. With the assistance of supportive findings, Goblet anticipated lecithin structure, including choline, margaric acid, oleic acid, and phosphoglyceric acid. The phospholipid chemistry made substantial advancement with Thudichum, who isolated various phospholipid fractions and characterized them by means of their nitrogen-phosphorus ratio. He depicted cephalin, which is synonymously known as phosphatidylethanol discrete from lecithin by solubility properties. From cephalin fraction, he also isolated ethanolamine and deemed it as a putrefaction artifact of choline. Similarly, in the Institute of Physiology and Chemistry in Strasbourg, France, Baumann and Renault in 1913 described ethanolamine phospholipids. Sphingomyelin, which is in Greek word, means tight binding with the marrow, was also isolated and described by Thudichum along with its molecular constituent. The constituent of the sphingomyelin includes the fatty acid, sphingosine, choline, and phosphoric acid. From the studies of Thudichum, it has been concluded that phospholipids are the chemical soul of all bioplasm and center of life. By 1927, sphingomyelin, lecithin, and cephalin had been explained, followed by the isolation of phosphatidic acid from cabbage leaves by Chibnall. 1-acetyl phosphatide from beef heart by Fulgen was described in 1939. Phosphatidylethanolamine, phosphatidylserine, and inositol phospholipids from bane by Folks in 1942 and diphosphoinositidide in 1949. Similarly, cardiolipin from brain was described by Pangborn in 1944. Categorically, there are three categories of phospholipids. The first one is the glycerophospholipids. They are synonymously known as glycerophosphatides or phosphoglycerides. They are a group of the glycerol containing phospholipids. It implies a few imitative of glycero 3-phosphoric acid that contains at least one encyl or alkyl or acyl residues affixed to the glycerol moiety with a polar head, which is consist of a nitrogenous base, an inositol unit, or a glycerol. The glycerophospholipids can further be sub-categorized. The first one among these glycerophospholipids includes phosphatidic acid, which is hydrolyzed to yield one equivalent of glycerol and phosphoric acid. And the second category is lecithin. Lecithin contains fatty acid, phosphoric acid, glycerol, and nitrogenous base choline. Other categories include cephalin. It contains the fatty acid, the glycerol, the phosphoric acid. Nitrogenous base is ethanolamine or cholamine or the amino acid serine. The last category in glycerophospholipids includes plasmalogens, which on a hydrolysis yields one molecule of aliphatic aldehyde, glycerol, fatty acid, phosphoric acid, and nitrogenous base, ethanolamine or choline. Among the three categories of phospholipids, sphingophospholipid is the second category, which is synonymously known as sphingosylphosphatides phosphatides or phosphosphingolipids. These are the group for the sphingosine containing phospholipids. It contains a long chain base, glycoside moiety with a phosphorus residue. It contains sphingomyelin. The sphingomyelin is having a fatty acid, a phosphoric acid, and a choline moiety. The peculiarity of sphingomyelin is that it lacks glycerol. The third category of the phospholipids are alkyl phosphocholines. They are phospholipid-like molecules. Henceforth, they are categorized under the phospholipid category. The alkyl phosphocholines are group of for the esters of phosphocholines with a long chain aliphatic alcohols. Opposed in chain length, opposed in position, and unsaturation of the cis double bond. Abel in 1992 and its team describes the remarkable biological and therapeutic activities of alkyl phosphocholines. Among the complex lipids, glycolipids is another category or the type of lipids which contains a glycosidic moiety, and this glycosidic moiety may either be the galactose or glucose. In addition to the carbohydrate, the glycolipids contain fatty acid moiety, sphingosine as a component. Sometimes one or more phosphate groups are present in glycolipids. The long-chain derivatives of sugars restrain most frequently in bacteria and plants a glycerol, which is a diacyl glycerol backbone, and in animals it contains a ceramide backbone. A phosphorylated polysaccharide lipid complex or a sterol may also be found. Simple glycolipids are composed of a carbohydrate moiety linked to one fatty acid or fatty alcohols. A wide variety of glycolipids are present in prokaryotes, especially in bacteria, where specific glycopeptidolipids or lipopolysaccharides are also available. Commensurate with the structure, glycolipids have the following categories. The first glycolytic category is based on glycerol-based glycolipids. The glycerol-based glycolipids contains mono or oligosaccharide moiety connected to the glycerol or the hydroxyl group, alkylated or acylated with one or two fatty acids. They might be uncharged and referred as neutral glycoglycerolipids, having a phosphate or a sulfate group. Corresponding to their structure, glycerol-based glycolipids can further be subdivided into the following: neutral glycoglycerolipids, glycophospholipids, glycosulfoglycerolipids. Another type of the lipids are neutral glycoglycerolipids. Most commonly, this class contain one or two saccharide units which are linked glycosidically to diacylglycerol or glycerol. However, neutral glycolipids with 3 or 4 saccharide units are also reported. Being essential in algae, bacteria and higher plants, they are positioned in photosynthetic membranes and are also present in minor amount in animals. About 85% of neutral glycoglycerolipids like DG, DG and MGDG are present in photosynthetic membranes of all oxygenic photosynthetic membranes. These glycolipids represent the most abundant lipid class on Earth based on the innate load of photosynthetic organisms. In another case of Plasmodium falciparum, a protozoan which is responsible for causing malaria, the discovery of a plastid without galactoglycerolipids heaves the issue of trouncing the galactoglycerolipids during evolutionary process. In a number of plants, galactosyl monoacylglycerol with a variety of galactose moieties have also been uttered. There are two families of neutral glycoglycerolipids. The first, which is a non-acylated glycoside moiety containing compounds, and the second, the acylated glycoside moiety containing compounds. Now, the neutral glycoglycerolipids, among these monogalactosyl diacylglycerol and digalactosyl diacylglycerol are the most common one. In 1956, Carter et al. Stated the presence of glycoglycerolipids in the wheat flour extracts followed by its explicit alliance with thylakoid membranes of the chloroplast, which was further reported by Benson et al. In 1958. However, the accurate structure of monogalactosyl diacylglycerol and digalactosyl diacylglycerol was later explicated by Carter et al. In 1961. This alliance is epitomized by the algicide or herbicide commotion of galveston, which is a popular monogalactosyl diacylglycerol synthase inhibitor. The MGDG synthase is an enzyme which is responsible for the synthesis of monogalactosyl diacylglycerol. After the monoacyl diacylglycerol and diglycerodiacylglycerol, tri-galactosyl diacylglycerol and tetra-galactosyl diacylglycerols, which are the superior homologs of galactolipids, were also recognized, and they were recognized in the plant tissues. In 2003, the group led by Zhang elucidated the structure of trigalactosyl monolinolenylglycerol, which is a new glyceroglycolipid, has been isolated from Premna microphylla. The Premna microphylla is a plant used in the Chinese folk medicine. After that, large amount of linoleic acid having a ratio of 18:3 and an explicit trienoic acid ratio of 16:3 is present in both monogalactosyl diacylglycerol and digalactosyl diacylglycerol. In higher plants, linoleic acid is the lone fatty acid in monogalactosyl diacylglycerol, and hence these plants are known as 18:3 plants. Numerous fatty acids have been docked in monogalactosyl diacylglycerol isolated from a brown alga, Sargassum thunbergii. In most fungi, glycosphingolipids is the main glycolipids, and galactolipids have been found in small amounts. In monogalactosyl diacylglycerol, an unusual fatty acid was described from a marine diatom, Skeletonema costatum. They were revealed to be metabolized into aldehydes, which are short-chain, octadienyl, and octatrienol, that could have lethal upshot on zooplankton crustaceans. Heptadienyl, another aldehyde, was also known to be produced from the icosapentaenoic acid of monogalactosyl diacylglycerol. Two linolenic acid acyl groups containing monogalactosyl diacylglycerol have been depicted in fruits of Rosa canina, popularly known as rose hips, possessing anti-inflammatory properties. These inhibits the cell migration, which is unservingly correlates to the anti-arthritis activity of rose hip herbal remedies. Other reports revealed the anti-tumor promoting properties, oxygen forage, and virus counteracting activities of galactosyl diglycerides from different sources. In Thailand, the digalactosyl diacylglycerol isolated from Clinacanthus leaves demonstrated antiviral activity against herpes simplex virus. The monogalactosyl diacylglycerol extracted from Sargassum muticum, an invasive brown alga, has been revealed to display inhibition of the microbial growth, including the bacteria and the fungi, which are responsible for the antimicrofouling activity. In the leaves of Linon Eucetacissimum, a novel family of oxylipins named linolipins has been exposed. The esterified residues, which is either one or two of divinyl ether, ethernolic acid, and parallel oxylipins comprising have been alienated from the flax leaves, which are damaged by freezing and thawing, or inoculated with phytopathogenic bacteria. In petunia hybrid flowers, it has been observed that the DG-DG synthesis increases and the pistils, pollinic tubes include elevated quantity than any other floral organs. In bacteria, the glycosyl diacylglycerol possesses glycoside moiety. For example, D-mannopyronosyl, D-mannopyronoside present in Micrococcus, D-galactopyrronisyl, D-glucopyronoside in Lactobacillus, and D-glucopyronoside in Pneumococcus or Staphylococcus, and D-glucopyrronysyl, D-glucopyronoside, and D-galactopyronoside in Mycoplasma. Beta-D-glucopyronosyl and beta-D-glucopyronoside is present in gento-biacyl diacylglycerol in Staphylococcus. In Propionibacterium propionicum, a novel glycoglycerolipid enclosing an ether-linked alkyl chain at third carbon position of glycerol has been revealed. In 1992, Manka et al. Isolated a diglucosyl diglyceride with an incredibly exceptional diglucosyl structure, that too from Thermotoga maritima. Inquisitively, 2 moles of palmitic acid esterifies the glycerol moiety and acylated on the 6-hydroxyl of the terminal glucose by 1 molecule of decanoic acid. It has been well known that the glycolipids present in flour act as surfactants and affect the texture, staling, and the volume of the bread. Selmar and his team, working with the glycolipid, reviewed the role of glycolipids in bread making and have shown that monogalactosyl glycerides and its lyso derivative perk up the baking performance of the wheat flour. However, in 1998, Hamburg reported the presence of digalactosyl diacylglycerol monosaccharides in the kernels of oat. So, today we have learned about the types of lipid. Actually, lipids are simply classified into three different types. The simple lipids, which includes fats, oils and waxes. The complex lipids, which is phospholipids, glycolipids and proteolipids. And finally, the another type which is called as derived lipids, which includes hydrocarbons, steroids and vitamins. So in this module, we actually learned the first part of the type of lipids. The rest part will be continued. Thank you.